The region of the bacterial cell where we find the DNA is called the nucleoid. Cell plate would be E. Found in plant cells? Yes, but not animal cells. Animal cells we know have the cleavage furrow. Bacillus would be M. Rod-shaped bacteria. Meiosis. G. Produces haploid cells from diploid cells. Remember, we start with diploid cells, and the idea with meiosis is reduce it by half. Reduce the chromosome number by half, so when sperm and egg come together, we have a diploid cell again. So we go from diploid to haploid, and then sperm and egg get together, diploid again. And the offspring, diploid. And that helps maintain the chromosome number for the species from generation to generation. Coccus would be H, spherical or circular bacteria. Microbiology A, no worries there. Cleavage furrow, W. Animal cells, yes. Plant cells, no. Egg cells are haploid, so V works for egg cells. And autoclave K, that was a good one. Questions on those? Points each. Okay. Multiple choice. Passive transport, what about that? Well, it does not require energy, yes. That's the definition of passive transport. But continue to read on. C, moves molecules against a gradient, so low to high. No. That describes active transport, where you use energy. You go against a gradient, but you paddle upstream, you spend a lot of energy if you paddle a canoe upstream. D, moves molecules down a concentration gradient, low or high to low. Now that works. So go with A and D. Letter E is the correct choice there. Next page. Number 2. This is probably the most missed in all the multiple choice. One you just have to sit and think about for a minute. Some protozoans live in water, and we're going to see this coming up. So we're going into the protozoans this morning. Have specialized organelles called contractile vacuoles. These organelles continually eliminate excess water that enters the cell. Water always going in the cell, okay. The presence of these organelles indicates that the environment these organisms inhabit. All right, so here's our protozoan. And there's the structure called the contractile vacuole. And as water flows in across the membrane from the outside environment, this contractile vacuole captures it and then pumps it back out of the cell. Otherwise, what would happen to the cell? That water kept going in, and the organism never got rid of it. What would happen to the cell? It would burst, yeah. It would die, burst, yeah. The membrane's going to, like a balloon with too much air in it. It's going to burst. Okay, so water moves towards the higher solute concentration. So where's the higher solute concentration? Here. We'll depict that like little dots here. Lots of solute in there. Sugars, amino acids, ions, stuff like that. Okay, so this would make, if this is the higher solute concentration, this would be what? Iso, hypo, hyper.
It's hyper.
Hyper. If that's hyper, this is what?
Hypo.
Hypo. Okay, so the correct answer there is letter C. It's hypotonic to the organism. The environment they inhabit is hypotonic to the cell, to the organism. Number 3, most of you forgot about my little clue and how to remember this. Many of you did. In a glass of sugar or water, the solvent is the water. Remember when I did this on the board? Dr. Orr would be turning over on his grade right now. Solvent, what's the next letter in the alphabet? Water. There's your association. That's a slam dunk question. Number 4, chefs. So will the vegetables in cold water to restore their crispness. Okay. This demonstrates the chef's knowledge of osmosis. Why is that osmosis and not diffusion? Membranes, cell membranes of the vegetables, yes. Cell membrane, so we're talking about osmosis here. Number 8, large molecules, such as glucose, may enter a cell through what process? Facilitated diffusion. Remember the proteins, the big proteins for the big molecules. We need a door, we need a window, we need a protein embedded in that membrane in order to move these larger, not all that big, but larger molecules. And the rest were pretty good there. Questions on page 2. Page 3. 11 and 14 were just to see if you knew what happens at the end of meiosis and what happens at the end of the cell cycle. So meiosis, remember, means a lessening number of chromosomes reduced by half. So if we start with 50, this is question 11, we're going to end with 25. Each gamete, each sperm, each egg will have 25. For number 14, the cell cycle is just repetition. It's just a copy. So we start with 50, we end with 50. 18, human females, where does meiosis occur? Where does meiosis occur? Not in the eggs. The eggs are the product of meiosis. Where does it occur? It occurs in the ovaries. In the ovaries. Testes male, ovaries female. And the rest were fine there. Questions on page 3. Okay, page 4. Yeah, number 20, I thought that was an easy one. If you knew what an endospore was, you could get that one. What about bacterial endospores? They help the bacteria, specifically the bacillus type of bacteria, survive harsh conditions. So letter A there. And 23, which of the following characteristics describing bacteria is false? All right, they have walls? Yes, that's true. Prokaryotic? Yes. Possess DNA? Yes, all cells do. Letter B, possess many organelles? No. That's a eukaryotic characteristic, not a prokaryotic characteristic. So B doesn't work there. D and F, yes. Both describe bacteria. Reproduce quickly, form endospores? Yes. Questions on those last few multiple choice?
Can you do number 2?
We haven't gotten there. Multiple choice.
Oh, never mind.
Anybody? Okay. All right, number 1, short answer. 3 ways bacteria may gain access to the body. You could have said food, water, and air. That's a pretty easy one. Food, water, air, touch, sexual contact you could have thrown out there, insects, that sort of thing. Any 3 would have done. Number 2, what is condensation or DNA packing? The conversion of chromatin to chromosome. Somewhere in your response I wanted to see that. Conversion of chromatin, DNA in the chromatin form, to chromosomes.
So if we said chromatin to chromosome, would we have the whole 4 points or just half of that?
2. It's 2 parts to the question.
Oh, okay.
Okay? So chromatin to chromosomes. The second part of that, why is it important? It organizes the DNA, making it more easily separated. Assuring that when the 2 daughter cells separate, that each daughter cell will get the same apportionment of DNA. So it facilitates this equal separation, this equal division of the DNA. If this doesn't happen, it's going to be very difficult to equally separate the DNA. So each cell has the exact same complement.
So I said chromatin to chromosome and said organizing DNA, but
You need to say, yeah, organizes it. That's basically what it is. Chromatin, chromosomes. That's organizing. But what I need is, I needed you to say something that, like, it's important because it assures that each cell gets the same amount. Okay? You were close. I remember your response. You were close, but You weren't quite there. Wow, that's right off the study guide too. Look at that, right there. Point number 3. Point number 4. What is condensation DNA packing? Why is it important? Shame on you if you missed it. A couple of you didn't even respond to that one. Shame if you were not prepared. You could not have looked at that. You could not have prepared if you missed that one. I'm sorry. I don't mean to hurt anybody's feelings. I'm not picking on anybody. But I'm just saying, wow, it's all there. 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 I would have killed in my undergraduate career. I would have killed somebody for a study guide like this. You know how many my professors gave me during my college career? Boom, 0. It was suck it up. It was there are your notes. Deal with it. Right there. Got to use those. You got to know those back and forth. Got to formulate responses for these in your mind. Write it down. Have it in your mind so when you come in and you see a question like that, you say, wow, that's right off the study guide. Life is good. It's right off the study guide. Got to be prepared. Preparation, preparation, preparation. All right, questions on number 2. And one other thing. You pay attention to the 2 parts of questions. A couple of you missed the second part of this because I don't think you saw that part or you didn't address that part. So read the question carefully so you hit every part of the question if it's multiple parts. Okay, 3 ways bacteria benefit humans. I think that was right off the study guide too. You could have said nature's recyclers. You could have said immune system. They beef up our immune system, especially at a young age. You could have said food production. You could have said natural type of insecticides, these sorts of things. Any 3. Next page. 2 reasons why meiosis is important to organisms that reproduce through sexual means. The 2 things, 1, it produces variability. During the crossing over, variability is introduced into the gametes that are being produced, the sperm and the egg. We said the beauty of that is you might have siblings, but you share some of their genes, but you don't share all their genes because you look different. You're different individuals. So variability in the population is 1. The second is that meiosis assures that the chromosome number for the species remains the same from one generation to the next. You go from diploid cells to haploid cells, sperm and egg. When sperm and egg get together, haploid equals diploid. The number is maintained. We said if that did not happen, what would happen with every generation? What would happen to the number of chromosomes? What?
They go down.
They would what?
Would they go down?
They would what? Not go down. If meiosis did not occur, what would happen with the numbers of chromosomes every generation? It would double. It would double. You have to reduce it by half, and then the 2 halves come together to make 1, basically. If you didn't reduce it by half, it would double with every generation. That can't be. Questions on that one? Okay, number 5. You're driving along the Manassas Turnpike this time of year, or usually early spring. Sometimes you see it in the fall, actually. It hangs around for a while. You may notice that many of the pine trees have brown needles. This was caused by exposure to highway road salt that was sprayed on the trees from passing traffic. See this a lot along sidewalks too. You have evergreens planted and people walking and salt spreaders that maybe these people push along the sidewalk. These trees get hit with salt. In the context of diffusion or osmosis, only one is going to be correct. Explain why these pine needles turn brown from the salt exposure. If this is your needle, there's your needle, and it's being hit with a lot of salt on the outside, you're exposing that needle to what type of environment? Hypertonic environment. High solute concentration on the outside. So the water molecules are going to move towards the higher solute concentration, the water molecules are actually going to leave the cells of the pine beetle, killing many of the cells. So you get the discoloration. So that's osmosis. So osmosis, hypertonic. Supposedly moves to a hypertonic environment. Kind of like throwing salt on a slug. We talked about that in class a little bit. Same deal. Questions on that one? Okay.
You still get points for putting osmosis but not hypertonic? Right?
Yes,
Okay.
yes. If you threw osmosis in, but you really didn't say, what's going on, somewhere in there I was hoping to see hypertonic, yes. Number 6, Bt milky spore. Why are they better for the environment than conventional insecticides? The bottom line is they kill only the target insects. That's the bottom line. They kill only the bad guys. It's like dropping a nuclear bomb over a city where you want to kill a bunch of people, but you don't want to kill everybody. You only want to kill the bad guys. And that nuclear bomb kills only the bad guys, and everybody else walks away. It's kind of the way it is. So you spray a tree with Bt or you put milky spore down in the lawn, and it's going to leave a lot of insects alone. They won't be impacted by it. But it's going to get the bad guys, the ones you want to get. Okay? It's also good for the environment simply because it doesn't poison other creatures. It doesn't enter the water and things like that. But somewhere in your response, I wanted to see that only kills the targets. Unlike regular insecticides, you spray it, good insects, bad insects are harmed. Questions on that one? All right, last one, the plowshare tortoise. Only about 400 left in the world, unfortunately. Concern for the species is due to its low population size, little genetic diversity in the population. So why is this a threat? We talked about that at the end of meiosis. We said the problem with endangered species is that they have so little genetic diversity that the first big environmental challenge that comes along, a warming climate, a drier climate, a cooling climate, something changes in the environment, a new predator, a new disease, a new virus. They do not have the genetic stock or diversity to survive this new challenge. Some might have it, but many may not. And with only 400 left, the new virus comes along and challenges the plowshare tortoise. It might kill 275 of the 400. Well, that's not good. It's even less genetic diversity than we had before. And that's kind of how the way, many endangered species go. It's a slow, subtle, not so subtle, it's a slow decline like this. And at some point, you just don't have enough individuals anymore to reproduce viably, find each other to reproduce, and the species slips into extinction. So it's too bad about the plowshare tortoises. They're having all sorts of problems. They're found primarily on the island of Madagascar. Habitat loss is taking its toll, and collection. These idiots go out and collect them and then take them and sell them somewhere in the exotic pet trade. So don't buy exotic stuff. Take my environmental biology class, we talk all about issues with exotics. People, I need an iguana. What kind of love does an iguana give you? Or, I need a snake, a boa, or a lizard. Well, a lot of these, not all, many are bred in captivity, but some of these come from nature. So you're contributing to the decline of some of these species if you go for this stuff, if you create a demand for it. And all you have to do is look into the Everglades National Park to see the result of the illegal pet trade. What's happening in the Everglades National Park, South Florida? Burmese pythons. Big, badass snakes not native to this country, but they were brought in because, I want a python. And then these idiots get them and they get to be too big, and what do you do with it? Well, if you're living in South Florida, you take it and release it. Oh, let it go in the swamp. It'll have a happy life there. Now they're breeding like crazy, and they're decimating the local mammal, bird, and reptile populations there. Because these are the biggest, baddest predators. Next to gators, they're right up there with gators. Right at the apex of the food pyramid. They are the top predators. They pretty much got the run of the place, and they can pretty much eat whatever they want, even small gators. So now it's a huge problem. It's a huge problem in the Everglades. Many mammal and bird populations are doing this because of these Burmese pythons. It's a mess. It's a mess. So they regularly have roundups there where people go in, the National Park Service, Fish and Wildlife Service pays people to go in and kill these things, give them a bounty. But then you have the animal rights people coming in saying, we can't do this. Oh, we're killing snakes. No, we've got to do it. We've got to do it to preserve the ecosystem. They don't see the big picture. The big picture is the health, the protection of the ecosystem, not these snakes that don't even belong in this ecosystem. Doesn't work that way. Anyway, don't get me started on this. It's disastrous. Disastrous for the Glades. Pardon me?
So they should go try to reason with it and then see how that works.
Yeah, exactly. Exactly. Okay. So what about things? If you look at the bottom of the last page, I calculated your scores. So if the semester ended today, this would be your grade. We're going to have about 70% of your grade is decided. About 30% left in play. We're going to have the final exam, the last exam, be about 100 points. We'll have the lab exam, I don't know, 40-ish points, somewhere in there probably. Some miscellaneous work. So we'll say about 200 points, maybe a bit less, still in play. So if your score, I'd say, is sitting below a 60, you really need to seriously think about, I hate to bring this up at this time of the semester, but the reality is the reality. The 29th is the last day to withdraw from the course without penalty. Get the W on your transcript versus an F. You don't want an F. So you have to think to yourself here, okay, what grade do I need? What grade is acceptable? If a C- is acceptable, and you're sitting, I don't know, in the mid-D range, I'd say hang in there, try to blow away the finals, and grab that grade. But if you're sitting maybe below a 60 somewhere, and I need a B, I need to get into the nursing program with a decent grade, it's not going to happen. As much as I'd like to say it's going to happen and say hang in there, keep trying, at some point everybody has to face the reality. I've been in your shoes before. Trust me, I know. The worst thing you can do is kid yourself saying, I'll do better on the final. I'll get an A on the final. Well, I hope you will. But I've been in your shoes before, and I've kidded myself, and I've been sloped by it. Especially during my freshman year of college. So my point here is that look at the numbers, think about your situation, think about how much time you have to put into a final exam. If you're bogged down with 4 other classes, and oh my God, I got all this work to do. Maybe it's better just to cut your losses a little bit, drop out of this one or drop out of something, withdraw, and focus on what else you have, the other courses that you have. But it's an individual decision here. That's the reason I gave you the number there, just so you know where you stand. Some of you are sitting, C- range, C+ range. You can move those up yet. 150 points is a good chunk of points. But it's not going to move you from an F to a B. That's not going to happen. It won't, the math isn't there. But if you're sitting at a C plus right now, certainly moving into the B range is a real possibility. You can do it. Or if you're sitting A minus, move into the A range. Or B plus, move into the A range. You get the idea. There's still movement, a bit, positive. But big movement, no. Unfortunately. Big movement, no. Yes?
Is this including the paper? Like this grade, including the paper?
No, does not include the line paper.
Okay.
So that'll help a little bit, but it's only 20 points. Okay, it's like that when I think I've mentioned it before. It's like it's like baseball. Early in the season, you have 50 at bats and you go 4 for 4 one day. That your batting average jumps up pretty good. It's a nice jump in your batting average. But at the end of the season, come end of September, when you have 500 at bats or 450 at bats, and you go 4 for 4, doesn't move your batting average much because you have all these points already. So in a similar sense, we've got a lot of points accumulated already. So even if you score a really good score, if you're sitting with a 55 average and you score a really good score on the final, it's going to move your grade, yes, but is it going to make it jump into the B range? No, it can't. The math just doesn't work. Okay, so think about it. If you need to talk it over, I'll be glad to sit down with you. A couple of you have missed exams. That score does not reflect the missed exams. It did not calculate that missed exam into your score. You still have those to make up. Does not include a drop quiz either, but that'll be a small adjustment in your score. It's not going to be, oh well, once he drops my lowest quiz, I'll go from a C plus to a B plus. No, it doesn't work that way. Not enough points. Okay? All right, so I don't mean to be a downer here on a Friday before a holiday, but you need to know where you stand to make a good decision. And there's nothing wrong with withdrawing. I hate to see people withdraw, especially at this point of the semester. But there's nothing embarrassing, nothing to be ashamed of. Look at my undergraduate record. It's peppered with W's along the way. For one reason or another, I was doing horribly in the class, something happened in my life, I had to withdraw. It happens. It's better than the alternative being an F. You don't want that on your transcript. Okay? Think about it. And as I said, if you need to talk to me about it, please talk to me about it. All right. Well, let me have these back. If you have questions on your exam,